So hello to everyone. I would first like to thank Laura and the organizers of this wonderful meeting to let me share with you some of our last results. My presentation will be in two parts. I will start by showing what we have obtained on our new model of rat for creatine transporter deficiency. And then I will show you some first results of our first trial with the AAV viruses in this rat. So to start, does not work. Okay, so very rapidly as an introduction, just to remind you that creatine is not only very important, of course, for the regeneration of high energy phosphates in the cells, but it may also be a neuromodulator or even neurotransmitter, and it is one of the important osmolytes of the brain. You know that the blood-brain barrier has a very poor permeability for creatine, so it's difficult to enter creatine from the periphery, and the brain needs, at least for a part of it, an endogenous synthesis for creatine. The creatine transporter deficiency leads to creatine deficiency in the brain. In fact, you don't have any transport on the blood-brain barrier, so you cannot treat the patients by creatine. There is no other creatine transporter on the blood-brain barrier. And due to the dissociation of AGAT and GAMT within the brain, you may have in some patients GAA accumulation within their brain, which could contribute, for example, to the epilepsies that are observed. In our group, we work on the rat since many, many years, especially on the brain. So we decided to generate a new creatine transporter deficient rat, taking advantage of the recent apparition of technologies like the CRISPR-Cas9. And we decided to choose one of the mutations observed in the patients that was described as completely abolishing the transporter activity, which is this amino acid 389, and the mutation is a change from a A to a G, and it makes the tyrosine becomes a cysteine amino acid. And so I will show you our main results for characterizing this rat in this first part of the talk. Our rat strain reproduces very well. We obtained double mutant females, but we did not try reproduction with them, but with all the other mix possible, we obtained expected Mendelian ratios. Our rat is creatine deficient in the brain. You see here a proton MRS. In red, it's the knock-in rat, so the creatine transporter deficient, and you see the huge decrease of the creatine peaks as compared to wild-type animals here. In all the brain regions we have scanned, we observe 80 to 85 decrease of creatine in these knock-in rats. Another feature of creatine transporter inactivity in these knock-in rats is in the kidney, where we observe what is characteristic in the patients, the increase of the creatine over creatinine ratio in urine. You see here, it's almost at 0 in the wild type, and we have almost 7,000 millimole per mole of creatinine of this ratio of creatine over creatinine. So it demonstrates the complete inactivity of the transporter, and in that case, it is in the kidney tubular cells. Interestingly, we also observe an increase of the GAA, so guanidinoacetate acetate over creatinine ratio in our rat. We measured creatine and GAA in the blood and the CSF of these rats. We observed a decrease of creatine in the blood, in the knock-in, a small increase of GAA, and no change of creatine or GAA in the CSF of these rats. And this is also a characteristic of the patients. The CSF is not a good liquid to look for creatine deficiency in the creatine transporter deficient patients. Our rats, or knocking rats, show decreased weight gain. It's very similar as what is observed in the mouse strain as the one that Laura just presented before. You see here the acquisition of weight a long time. At 20 weeks of age, it's about half the weight of a wild-type rat for the males. We also looked at all the other metabolites that could be measured by the MRS. We are using high magnetic field MRS at 9.4 Tesla. You see here the whole list that can be measured. What is interesting is that in contrast to the decrease, of course, of creatine and phosphocreatine, we observe a quite strong increase in glutamine, glutamate, and N-acetyl aspartate in all the brain regions we have measured. And especially for glutamine, we wonder whether this could be a mechanism of osmoregulation to counteract the creatine decrease in the brain of this rat, to maintain a kind of osmotic pressure homeostasis. I already showed this data of histology last year in Rotterdam, but just to show them again, what we are doing a lot of histology in the brain, and now these are results in the cerebellum, where we have observed a thinner molecular layer, decreased expression of MAP2, phospho-NFM, neurofilament of medium weight, and the total NFM. So and nothing changed on the MBP genes, which is expressed in oligodendrocytes. So these are proteins that are for MAP2 in the dendrites, for phospho-NFM in the axons. So our knocking rats may present cerebellar connectivity problems, and we are now analyzing the whole brain regions to look at more details on these neuronal defects. We did not see much changes in the number of neurons, so probably we mostly affect the fibers of the neurons, and we don't see much changes in the microcapillaries within the brain parenchyma of our knocking rat. Finally, we see some changes in the astrocytes, and this is also in this cerebellum. You see here in the knocking rat in the granular layer, we observe an increased number of astrocytes and a kind of altered morphology of the Bergman glia in the molecular layer. So maybe astrocytes are affected in these knocking rats. Finally, we also did a lot of behavioral tests, and to be short, I show you two figures here. We observe more grooming behavior and impair the working memory in our rats, and a systematic tendency in decreased sociability in our rats. So these are some of the results we have obtained. Lara Durand-Trio, who is postdoc in my group, is now working a lot to characterize more deeply this knocking rat for creatine transporter deficiency. So as the first intermediate summary, our knocking rats are creatine deficient in the brain, no creatine transporter activity in the kidney, decreased body weight gain, increased glutamine, glutamate, and N-acetyl aspartate in the brain. So maybe osmoregulation problems in these knocking rats. Morphological alteration of neurons in the cerebellar cortex, and some autistic features observed in the behavioral test. So we consider our SLC6A8 knocking rat as also a kind of promising tool to better analyze the disease and try to treat it. So now I turn to the second part of my talk with the challenge of treating SLC6A8 deficiency. I also showed this slide last year to remind you that to make a molecule enter the brain parenchyma, it has to cross at least 5 membranes with 3 uptake mechanisms and 2 release. So it's quite difficult to make a molecule enter the brain. What we have chosen to try to treat the SLC6A deficiency in our rat is to use the adeno-associated virus technology to try to reestablish a functional transporter, maybe at the blood-brain barrier and within the brain parenchyma. So I will share with you very new slides that Gabriela Fernandez, who is PhD student in my group, has obtained very recently with our first trial of AAV injection in our rat. As every one of you, I guess, we also took a lot of retardation in our work due to the COVID crisis, but still we obtained these first nice results. So to try to reestablish the creatine transporter within the brain of our knocking rat, we selected AAV9 serotypes. This first experiment that I will show you is in wild-type rats, trying to use a vector with the same promoter as the one we will use with the creatine transporter, but with the GFP protein, to look at which kind of brain cells are transduced, how long they maintain the expression of the gene, in that case the GFP, the proportion of cells, the identity of the cells that are transduced. And we tried two modes of injection, intravenously in the tail and intracisternal, so intrathecal within the brain. And we checked two different doses of viruses within these rats. So I will show you 4 slides with these first results. This is the expression of the GFP protein 5 weeks post-injection. We inject the young rats at 11 days postnatal. And you see in green the expression of the GFP protein within the cortex, the hippocampus, corticospinal axons, the huge expression of the GFP, and neurons of the dentate nucleus, Purkinje cells within the cerebellum. And this is by intravenous injection. This is the same, but with intracisternal injection, 5 weeks post-injection. You see the nice expression to very high number of cells within the cortex, hippocampus, the same in the corticospinal axons, cerebellum, and the dentate nucleus. And then 16 weeks post-injection, so 4 months post-injection, the same experiment, intravenous injection. You see 4 months after the injection, we see very well the GFP expression in the same regions as the one that were observed at 5 weeks post-injection. This is intravenous, and this is intracisternal, and it's the same. You see here a nice Purkinje cells that is transduced by the virus. So let me stop here my presentation of our results. We are now doing the next experiments with the vector, with the transporter, and we hope that in the next month we will have gone from creatine transporter deficient rat to the correction of the deficiency in these rats. And to prove this, we will do MRS to see whether we have creatine reestablished in the brains, and of course also through the behavioral test and the measure within the blood and the brain histologies to look at the correction or whether we have corrected the creatine transporter deficiency. Thank you very much for your attention. I would like to thank all the collaborators of my group and other groups in Lausanne, the Brain Mind Institute, the Center for Biomedical Imaging, and the clinical neurology in the CHUV. The work I presented is essentially the work of Lara Durand-Trio, who is a postdoc, and Gabriela Fernandez-Pires, who is a PhD student. And of course, I think the funding agencies like the Fonds National Suisse de la Recherche Scientifique, and you, of course, ACD, for letting me present you our data. Thank you very much.
Thanks so much, Olivier. This was fabulous. And I think we started this day on a great note, like a lot of new results, and results we're all very excited about. So we're going to invite Dr. Nicola Longo, who's the head of our Scientific and Medical Advisory Board at the SMAB, to moderate the session, together with Carol Sheoa, who's our close partner at Extraordinaire. And let's make sure that both Carol, there we go, and Laura can unmute as well. Perfect, and I'll let you take it away. So the goal here is to go through some of the Q&A, add your own. Dr. Longo and Carol are gonna take turns picking some questions. We had lots of good questions coming in.
I want to thank both speakers for the excellent presentation. And the main thing is that there were a lot of new data that were presented. There are already about 20 questions that already have flown in. And I will try to summarize some of them, because not all of them are, some of them are repeated. And the co-moderator, do you want to introduce yourself a second?
Hi, Laura, and hi, Olivier. Thank you very much. It was very interesting, maybe a bit technical for people like me, not a physician. If I can go ahead with one of the questions for Laura. Someone is asking that you mentioned that your gene therapy work has been delayed by COVID. Is it on hold until there's a widespread preventive or treatment for COVID, or do you think it could resume sooner? And what type of gene therapy are you working on? Is it development of gene therapy for CTD or a precursor?
Well, no. We are now ramping up again with the lab, so we are starting to do experiments on the gene therapy project. So we are now running on it. So we hope that no more delay will be caused by this pandemic. And the gene therapy project rely on AAV, adenovirus, adeno-associated virus, very similar to that presented by Olivier. So it's the AAV9. And the idea is to investigate different routes of administration, such as the intraventricular, the intrathecal, and the intravenous systemic administration. So it's yes, it's a development of gene therapy for CTD.
Laura, while we have you on, there were a few questions about the parvalbumin Cree mice. How do they actually perform in the Y maze? The second question is that obviously those are specific neuronal cells that you see in mice. Now in human, do we see degeneration of specific area of the brain as well? And the last question is there any neurodegeneration in your animal model of creatine transporter deficiency? There are three questions in one.
Okay.
So,
for the first question, we did not see any. No, sorry. We have found a significant decrease in the spontaneous alternation in the Y maze. So this suggests an alteration of the working memory in these parva-Cray mice, very similar to that observed in the ubiquitary mouse model. For the other two, which are actually very good questions, in our mouse model, we do not find any kind of neurodegeneration, at least at the cortical level, while we found at least a slight decrease in neurogenesis in the hippocampus of adult mice, and a slight decrease in the hippocampal volume of adult mice. But at the cortical level, no sign of the neurodegeneration, both at the level of excitatory and inhibitory neurons. As such, for the different regions involved, we are now investigating this issue more in detail, and at the first look, I would say that the answer is no. I mean, different brain regions could be differently affected by creatine deficiency, because we found that, for example, the visual cortex is less affected with respect to the prefrontal cortex. I guess this could be probably related to the different timeline of development of these different cortical areas, because, for example, the visual cortex develop sooner than higher associative cortical regions, and maybe creatine is coming from the mother is enough for at least the first development of visual cortex. But these are very preliminary data, and this is the reason why I didn't present any of them.
Thank you. Dr. Braissant, so the first question is, you described that there is an accumulation of GAA, an increase in GAA in plasma. What do you believe is the mechanism for the increase of GAA? And I don't think that we see that very much in human patients. Am I correct or am I wrong?
No, I think you are correct. In human patients, we don't observe much increase of GAA in the plasma of the patient. In our case, it's very small. It's significant, but it's very small. If you looked at the doses, we go from a 5 micromolar to 10 micromolar. So it's not much as compared to the decrease of creatine that we observe. We don't know precisely the mechanism so far. Maybe it could be due to what we have observed in the brain, but could be also the case in other peripheral tissue, is that the creatine transporter is also able to take up GAA. And in case of inactivity of the transporter in our knock-in rat, GAA remains at a somehow higher level within the blood, but we have to demonstrate it. It's speculative so far.
Thank you. The second question, everybody got very excited by your gene therapy, and there are 25 questions about it. Let me summarize a few of them. So the first question is it painful to inject the virus, the intracisternal injection? The second thing is, what is the dose that you use for the IV injection versus the intracisternal injection? Let's start from these two.
Okay, so the dose are 10 to the 12 or 10 to the 13 virus particle per kilogram for the intravenous injection, and it's 10 times less for the intrathecal or intracisternal injection, so 10 to the 11 or 10 to the 12 viral particles per kilogram for the intracisternal injection. What I have shown you here are the highest doses. So we had to stop everything during this spring, so we could not inject the low doses, but this has just been done by Gabriela 2 weeks ago. So we have now rats with the low doses that we'll observe in a few weeks. And the first question was, is it painful for the rat to be injected?
Correct.
Okay, so we do it under anesthesia. We don't observe any stress post-injection so far. So, but it's difficult to ask the rat if he feels pains. But you see, the intravenous injection appears to work also quite well. So we did the two ways of injection to compare and to choose after which one would be the best.
So just from my clinical experience, now we use intrathecal injection for SMA gene therapy. The second thing, we have several diseases called lysosomal storage disorder, where we do a spinal tap about once a month. Third thing, there is an approved enzyme replacement therapy for a condition, for a type of neuronal steroid lipofunctionosis, in which they place a reservoir where they can do repeated injection. One of the things that people need to understand, that gene therapy would be a one-time dose, so it doesn't need to be repeated multiple times, and that would be one of the big advantages. One more question while I have you here. What type of toxicity are you going to look at? And the second question is there going to be an immune response to the injection of the virus?
Yeah, so this is the definitive test that we have to do. Some of these AAV strains generate a kind of immune response. So everything has been taken, but we have to check this in the following weeks, now that the lab is starting again slowly. We will also look at toxicity like cell death, whether we generate cell death through the AAV injection. So a whole battery of tests to see whether our rat support or the injection. Yeah.
I have a more general question on gene therapy. What does the pathway to approval look like for gene therapeutics, and does it vary compared to traditional medicines? Do you see more hurdles to get it approved?
That is a good point.
So I'll interject for a second. I think we're going to hear a little bit more about that too for the parents. I'll let Dr. Braissant give a brief, well, please feel free to give a brief answer, but we'll get into more details to educate our parent community too. So hold on, folks. We'll talk more about it later too.
So honestly speaking, for my part, as a very fundamental biologist researcher, I did not get into these administrative pathways for approval for human gene therapy. But now there are some AAV solutions that are given, that have been approved. So it is possible, definitely.
Okay, and also a question about your latest research, Olivier. Should we assume this research is at least years away from a treatment for our children, or could it be sooner? How optimistic are you?
Well, I'm very optimistic for my rats, and I hope that in the following months we will see something and hopefully reestablish the creatine transporter in our rats. But of course, to transpose this to the patients, it will take quite some time more, probably, yes, to have the right preparation with all the yes, the toxicity test, the purity, all these kind of things. It's not an easy way. I would like to at least to make the proof of concept, and then we will see.
I would like to move back to Dr. Baroncelli, and the question that had been asked: how does your mouse model differ from the one of Dr. Skelton in terms of gene knockout? That it is the first.
Okay, well, basically they are both knockout mice with the deletion of the SLC6A gene. Basically, we just targeted different exons for doing the same thing. The second mouse model I showed you presented conditional deletion of the transporter gene only in parvalbumin neurons. But this is just a model we are using for investigating the pathological mechanism of CTD. The ubiquitary model is definitely the best one for the translational studies.
The second question, that you have seen abnormal neurovascular coupling in your animals. What are we supposed to see in humans who have creatine transporter deficiency? What would be the correlate of what we are seeing in mice in humans?
Well, basically, it's something that we are trying to do now. So we are using these functional NIRS, which is a technique very similar to IOC imaging in mice, and is a CAP-based technique, non-invasive, using infrared light to study hemodynamic changes in the brain of children. And we would like to understand whether we found the same hemodynamic changes in CTD children as well. The meaning of these findings, well, I think that they could be twofold. From one side, basically, if we have a different hemodynamic response in abnormal hemodynamic responses in CTD children, we should take into account this aspect when we are doing fMRI experiments, because if there is a neurovascular decoupling in these children, maybe the responses of fMRI could be different with respect we expect. And from the other side, we could use hemodynamic changes as biomarker for the follow-up of the disorder, and also for investigating the response to therapeutics in children, because it's somehow can predict the brain function in this population.
Thank you. So the other question is that you have shown some results about the EEG in your animal and also in humans, and they wanted just you to repeat a little bit what did you actually find on the EEG, what that actually means.
Well, basically, what we did is to analyze the spectrum of the EEG, so the different band of activity oscillations in the brain of mice and patients. And what we found is that there are at least two range of frequencies, the so-called theta and alpha band, which are altered in the brain of mice and patients with the CTD, with a lower power activity of these bands. And these could be another very useful biomarker for following up the disease progression, but also the response to therapeutics. And moreover, we have seen that at least in mice, we have a very low numerous sample for humans. So I'm not sure about that, but we have seen that at least in mice, some sleep issues could be present. So we are planning to perform some more investigations on this point.
There's a question from Matt Skelton for Laura. Okay, I'll read it. Hopefully I'll read it well. Did you test the parvalbumin Cree mice in the Y maze as well?
Yes, we did that, and we found this lower spontaneous alternation, so a deficit in working memory.
And another question about cardiac manifestation in the mice. Do you see any potential reversal with gene therapy?
It's for Olivier, I guess.
Yeah, for Olivier. Yeah, for both, but.
Once again, the question, cardiac problems, do
you see any cardiac manifestation in these mice and potential reversal with gene therapy?
So we did not look at it so far. Our rats, it's not mice, but our rats appear quite well, but we have to check the periphery. We concentrated on the brain a lot, but we will check this, yes.
And Laura, on your mice, do you see
Same for us. We just seen decreased levels of creatine in the heart of these animals, but no other investigations, because our main expertise is on the central nervous system. We are now collaborating with another group very close to our lab for cardiac phenotype, but we have no results so far.
Okay, thank you.
Thanks, everyone. I think this was a fabulous conversation. A big thank you to our amazing moderators. I think we'll go to a quick break for 5 minutes before the next session, but a big round of applause to both the speakers and Dr. Longo and Carol for doing a fabulous job moderating this. And we'll send the questions to the speakers too, so that you see the wide range. Thank you so much for all your hard work.
Thank you.
Bye.